A quick overview of the California Conservation Genomics Project. It's a project that we started in 2019 was when the project was first funded. We really got it off the ground probably 6 months or so into 2020. So it's been going for, getting on close to 5 years. And we are in the final phases of it. And it's my real pleasure to report to you how we've been doing. So remember that the concept of the California Conservation Genomics Project is to complete a very large empirical project that includes a large number of species, which we're sampling at the, very densely at the within species level across the state of California. And the goal is to use this multi-species, multi-individual data set to be able to look for patterns of genetic health, genetic connectivity, a variety of different important genetic topics that go across the entire state and basically provide a genetic roadmap of the state's diversity and connectivity to decision makers who can use that information. And what I want to do is just give you a quick summary of it. So our initial goals in the project, we had 235 species identified across 153 genera or projects. And those are our sort of primary units of analysis, those 153 projects. Associated with each of those was a reference genome. 6 of them had reference genomes. So we had to complete 147 new high-quality reference genomes with RNA-seq data for annotation. And the goal was to roughly within each of those 153 resequence 150 individuals using short-read data mapped back to those high-quality reference genomes. We originally had targeted a total of something a little short of 22,500 complete genomes that we would be resequencing, and then have our own variant calling and quality control pipeline and analysis pipeline. So it's the final year. Where are we at? How are we doing? In terms of reference genomes, as I said, the goal was to complete 147 reference genomes. 6 already existed that we felt we could use that were up to our standards. And where we're currently at is that 143 of those 147 are finished. 134 of them have been released on NCBI. 135 submitted, so there's one that's submitted that's not released yet. Of those, 3, we would just have to say have basically failed. We just could not get them to work for whatever reason. And we can talk about that if you wish, but it gives us a success rate of about 98%, which we feel very good about on the reference genome side. In terms of annotation for those reference genomes, investigators could submit tissues if they wished or not, or if it was convenient. 107 of those 147 projects submitted a total of 633 tissues. All of that RNA-seq has now been collected. All of it has been submitted to NCBI. And then NCBI runs it through its standard pipeline of annotation pipeline. 11 of those, as of a few days ago, were completed. And NCBI is trying to kind of chug through those for us. So that's feeling pretty good as well. In terms of the resequencing, which is really the heart and soul of this project, not the reference genomes. As I said, we had 22,443 proposed individuals to resequence. And these maps just show the proposed distribution of those for plants, inshore, vertebrates and invertebrates, terrestrial invertebrates, and terrestrial vertebrates. Of those 22,443 proposed whole genomes, about 14,000 of them were scheduled to be completed at our core facility that we set up here at UCLA, and about 8,000 roughly were going to be completed by individual principal investigators and then submitted to us for analysis. Of those, we've completed, again, as of a few days ago, about 9,000 of the 14,000 at the mini core. We're probably up to 9,500 by now. All of the ones by the individual PIs are done. For the ones that we analyzed down here, we had to drop about 400 of them because of data quality issues. The principal investigators came up about 2,000 short. I think they just either didn't work or couldn't get the samples. So where that leaves us right now is very roughly about 15,000, perhaps a bit more, are completed. And we expect a total of about 20,000, so pretty close to our original goals. In terms of analysis, 65 projects, and you're going to hear more about analysis from other speakers in this session. But 65 projects are completed through the variant calling pipeline, and all of those have been run through our basic landscape genomics pipelines that you'll hear about. And then our genomic offset analysis, which is one of our, sort of fancier, more computationally expensive analyses, has been completed for aid, and more of those are sort of chugging along as we speak. So we have, roughly speaking, half or a little under half of the projects completed all the way through, and we're rounding out the rest of them as we go. Bumps in the road. People always wonder, what could possibly go wrong? Well, just about everything. There are lots of bumps to choose from, and we've certainly learned an awful lot in the last 5 years. On the reference genome side, I think the biggest ones are that a lot of genomes are just way bigger than we thought they were, some by an order of magnitude. We thought they were 1 gigabase. It turns out they're 14, this kind of thing. And it's just the price of doing business with very, very understudied taxa from across a vast region like California. A few species just won't assemble. I don't know why. We've had real trouble with decapod invertebrates. Crab and lobster, really tough. I don't know what it is. We haven't been able to figure it out. We've had at least one example of a species that we thought was diploid, turns out to be polyploid. Well, that's a big issue. And then there are some composite genomes. Lichens, which we have 2 or 3 of, are the biggest ones, where there's, a fungal and an algal associate, and trying to get both of those and sort that out bioinformatically is really tough. On the resequencing side, it's certainly lessons learned. Assemble all of your tissues before you start. We have many projects where they've kind of trickled in, and that causes a lot of headaches. It's really great to have all of them before you start. Insist on high quality material, because low quality material just really clogs up the works. And I think a rule of thumb is the greater the taxonomic diversity, the more headaches that you have. And the final frontier, and this might be something that would be fun to talk about at the end of the session if we have time, is our interactive user interface. So we're going to have all of this data in all of these maps. We are working with a company called Green Info Network to create an outward-facing interactive set of maps and analysis tools. This is for managers. This is for consumers of these kind of data like yourselves. We're just getting started on it, and we'd love to have some input. Acknowledgements, there's the team. You're going to be hearing from a number of them. There's many other people, obviously, who have contributed, but this is a lot of our kind of core team. And then just to say, you can also see this in the chat, but the way we're going to work the next set of talks is that Eric and Ann, 2 postdocs who have worked very, very hard for a couple, 3 years on the project, are going to be giving talks with a Q&A afterwards. We'll then move on to 5 lightning talks by Kay, John, Kim, Nathan, and Anusha. All of those were just going to be done sequentially. And then at the end, if you can stay on, we should have plenty of time. We can have a discussion of any and all things that you might want to talk about. Although we will have individual discussions after Eric and Ann's. So I will stop sharing there and turn it over to Eric to take it away.
How are we doing, Brad? Can we see the slides?
Yep.
Awesome. Well, thanks for that Great introduction from Brad on the project. I'm excited to be here to chat a little bit about some analyses that we're working on. And for this talk, really focusing on how regional conservation genomics can give us insight into the factors that affect population health. And there we go. So as you know, many of those who are here know really well, it's a pretty exciting time to be doing genomics in the conservation space. It's really something that's fully entered our conversation about conservation, and this is realized by things like news articles talking about conservation genetics, or the UN setting goals of things like protect 90% of the genetic diversity of populations sometime in the next, 10 years or even less. And so this is, already a field that's having an impact and growing, and with it, it also comes with some growing pains. And that comes from, as scientists, if we look around the literature, we see things like heterozygosity is great for protecting tortoises. It seems to benefit survival, and we should protect it for conservation. Followed up by other studies that are like, well, maybe there's an inflated significance of neutral genetic diversity and conservation goals. And I see all of this as being a little bit part of, the growing pains of how do we apply genomics and genetics to conservation challenges. And part of this comes from my perspective at least, the different ways in which we might measure and quantify genetic variation. And so fortunately, things like population genetics offers us a lot of insight into what the kind of metrics we measure, where they might give us insight and how they might be used. And I'm showing sort of a high level, bit of a hand wavy diagram to give some insight of what I mean. And so we can take measures of things like historical effective population size that give us some insight into demographic change in the past. And oftentimes when we measure this, we measure something about major ecological shifts that happen sometimes in the past, like glaciation cycles, for example. If we measure local patterns of genetic diversity in populations, we're often capturing things about historical population change. And this might be somewhere sort of in the mid to long-term past that this data is capturing. If we take measures of things like inbreeding, we can often get snapshots into very recent effects if a population has gone through a recent contraction and individuals are breeding with close relatives. And so you can see that sort of from a high level, there are different measures that we might take that tell us something about genomic health, but each of them might have different factors that have led to the estimates that we get in current time. And these are important distinctions to make, because if we think about how genomics informs us about extinction probability and the conservation of populations, really the genetic health of a population is something that can contribute to these extinction vortex processes. So as populations experience declines, no matter the factor, as populations become extremely small, things like the buildup of deleterious mutations and the loss of genetic diversity can then exacerbate and contribute to these declines happening even faster. And so it's important to understand these metrics, but it's also important to understand what are these sort of relevant timescales and what are the factors that we can actually learn from different measures of genetic variation. Now, we've already had a little bit of an introduction from Brad, so I won't spend too much time on this. This project that we're going to be talking about in the session today, the California Conservation Genomics Project, it's a large-scale project. We've got something like 20,000 samples of about 150 taxa. Each one of them has been sampled across the state of California. And something I really want to emphasize that's powerful about this data set in California is that we have a whole lot of different taxa. We've got plants, we've got animals, we even have all these sea creatures of a variety of different taxa. And so it's an immensely taxonomically diverse group, which provides a lot of opportunity for us to ask how these different measures of genetic variation inform conservation both at regional scales and local scales across the state of California. And so we're going to, in this talk, focus on these three sort of common measures of genetic variation. One of those is effective population size, on which we can estimate with genetic data, and we can make estimates for the history of effective population size over time, as in this example down here, where you can see that over time, the population of this species has declined towards the present. And we can also mark different cycles, either glaciation cycles or environmental change cycles that happen over the course of time during those declines or during those expansions that you can measure with this. We can also measure nucleotide diversity, so overall genetic variation within a population using a measure of pi if we have many individuals that we can calculate this for. And we can also estimate metrics of inbreeding. And particularly with genomics, we have a lot of power to measure recent effects of inbreeding, because as we see in this example in this slide, we've got heterozygosity plotted across a single chromosome. We get to this area where there's very low heterozygosity, and this is a long run of homozygosity. And so it's a track that's a signature of very recent inbreeding between related individuals. And so for each individual, we can quantify what fraction of the genome is contained within these runs of homozygosity. And this can be a rich, there's a lot of information here because over time, these runs of homozygosity will break down due to recombination if there's outbreeding. So they can also give us a temporal component into when we think inbreeding happened. Now, each of these factors are going to influence mutation load in different ways, as well as the ecological and evolutionary relationships amongst different taxa and the traits that these individuals have. And so the goal of this project is going to be to ask how do these different factors both affect each other and how well do they predict mutation load, which can be one way in which we can think about where the boots really hit the ground in terms of the effect, our ability to infer something about population health. So the first thing I'm going to show here is really a snapshot of species level variation in inbreeding and nucleotide diversity. So over on the left on the y-axis, we've got all the different taxa that we have in hand so far. It's something like 65 different species. And within it, I estimate what portion of the genome on average within these populations do we see. That's an individual dot and some confidence intervals. And what you can see is that there's quite a bit of variation. We have down here, species have very little levels of inbreeding, and other populations where we have individuals that have as much as 30 to 40% of the genome is contained within these tracts of homozygosity. Now, if we compare this to nucleotide diversity over here on the right, what you can see is that they're not really particularly strongly correlated. We have a relatively small correlation coefficient between these two things. And so just by looking at nucleotide diversity, which is often done in conservation genetic studies, doesn't necessarily give us this level of insight as we can gain from looking at these rows. Now, if we zoom in a little bit more, you'll notice that for each species, we have multiple different points for each one. And this reflects something that I think is really powerful in this data set, is that many of these projects have multiple subpopulations within them. And sometimes if we see strong differences, like we see here, one population has relatively low inbreeding, another population has relatively high inbreeding, that might be a strong metric that one of these populations is experiencing disproportionate declines relative to the other. And now we see some old trends that have been studied pretty well in these outliers in this data set. We have mountain lions up here, which have relatively high levels of inbreeding due to recent population bottlenecks. Spotted owls are one example here that also is a species that is declining rapidly, and we see some high signatures of inbreeding. But then nicely, we also have these extremes on the far end, things like trees that seem to be relatively outbred, where we don't see this sort of buildup of inbreeding. And I really like this because it allows us to kind of polarize these examples against these extreme examples of population decline to those that are quite outbred. Now we can line this up with these measures of long-term effective population change. So over here, I'm showing the puma, which you can see time kind of going towards the left, have experienced this recent and rapid bottleneck, which has likely contributed to this decline in these populations, in contrast to this tree species, which has experienced some moderate declines in the relatively recent past, but not going down to very low numbers and also recovering recently, which is likely why we're not seeing this signature of a bottleneck in this other metric. Now, we can also ask, what's something that we're interested in is regionally, do we see areas of California that might be experiencing similar population declines across species? So we can aggregate these statistics across all these different populations by ecoregion. So over here on the left, I'm showing the average fraction of inbreeding across a lot of different species in the state. You can see sort of by eye, you might pick out the Mojave Desert, for example. There, we're really sparse on sampling still, so I wouldn't necessarily really overthink that. But maybe we see the coastal populations in some central valleys, we see sort of elevated values. It's less dramatic in genetic diversity, but we still see some potential patterns there. Now, what's really powerful about this data set is we can look across all these different species and ask if there's common trends. A really simple model is, for example, does ecoregion or your species identification better predict how inbred you are? Overall, what you find with this is that what species you are is a better predictor of how inbred you are than ecoregion. I don't think this is too surprising. It would take sort of the whole region have gone through some systematic, massive decline across all different species to see a signature here, but it does tell us something about how the ecology and evolution of the different species is likely contributing to the variation that we see across taxa. Now, I also want to link this in with mutation load, which is this process of the buildup deleterious variants. The first step there is to identify deleterious variants, which can actually be quite challenging. There's kind of two general methods we could do this with. One of them is to identify coding mutations that alter the amino acid that's coded. Now, this requires a really high-quality genome annotation, which is one thing that makes it challenging. Another way to do this is to align a lot of genomes and then look for positions across a diverse array of different genomes and say, if a position is heavily conserved, that's likely something that if you change it, it's likely deleterious. This is challenging because you need a lot of reference genomes and you also need to align them all to calculate these. we'll start with coding mutations. In this example of a songbird that has two populations in California, you can see that we estimate that one of these populations tends on average to have more homozygous deleterious sites. So these are deleterious variants that we inferred as non-synonymous mutations that are likely exposed to selection because they occur in the homozygous state. And so on average, we see a difference here. But if we calculate a statistic that accounts for divergence time between these two different populations, we see that these two populations don't differ significantly overall in the relative mutation load. This is so not an exciting example, but it is one that we have an annotation for today, and so we're readily able to calculate these statistics for. We can also see that if we compare this measure of load against this measure of inbreeding, we see that inbreeding is a relatively good proxy for how many homozygous deleterious sites these individuals have. And this may be nice because it's quite a bit easier to estimate inbreeding than it is to estimate mutation load. And so far, we're kind of talking about population scale metrics, but we can also think about individuals. And if you look across the state, then one general pattern you observe in this taxa is that kind of on the edges of the ranges tend to be where we see individuals with higher mutational burden. And so this may be a bit of an edge effect. We also might see a little bit of a population stratification effect that leads to this. And so I think this is a really exciting pattern to explore as we expand this across different other taxa. Now, a challenge with those coding sequence evolution is that we need high quality annotations, and this is going to be a challenge for a lot of taxa on this project in the short term. And so another approach to do this is to align genomes and calculate these scores ourselves. So we're starting to do that in our lab here. I'm showing an example where we've assembled model organism species, all the CCGP species, and some other examples to then align all these genomes together and calculate conservation scores for each site. This is a bit of a computationally challenging task, but it's something that we've worked out a really nice pipeline for here at UCSC. And what we find so far is that if we take this example from this bird a moment ago, we find that those deleterious sites also have very high conservation scores from this measure of aligning genomes together, this phylo-P metric that I'm showing here, where we see a nice, the deleterious variants tend to have higher scores as you would predict in the direction of this metric. So this is great for the species we don't have annotations for yet. We're going to be able to use this method and apply it to this kind of analysis to be able to jumpstart those comparative methods that are really exciting about this project. So I'm going to wrap up there. A few takeaways we have so far. Genetic diversity in and of itself, it isn't particularly strong predictor of recent bottlenecks. Inbreeding is giving us some good insight into that, and it seems to also be a good proxy for mutation load. The next step is to really expand this across the different taxa. This requires a little bit of heavy lifting on the computational side, and I think we're going to be set up well to handle that now. And then from sort of the comparative analysis I've presented so far, regional effects seem to be relatively minor to the species-specific effects. And the next steps are to really bring in additional ecological and trait data to see what are the things that predict both individual and population level variation in these traits. And that's really exciting. We've got a huge taxonomic diversity to take advantage of here. And it's going to be really exciting to see what are some of the general patterns we get from this. As you'll know from the session, this is a huge collaborative project. It's a core team of folks who have been involved. There's some really key collaborators in here, like Ann Chambers and Anusha, you're going to hear from pretty soon here. And of course, the Corbett DETIC lab, which has been key to getting this off the ground, especially Ann Nakamoto, who's doing this great work generating these alignments of all these genomes to enable this mutation load analyses. So with that, I'll wrap up and take any questions. Is that what we're doing? We're doing questions, right? Maybe.
That's right. That's
okay.
right. So we're a couple minutes for questions, 2 or 3 minutes if there are any questions from the audience. You can speak up, or you can stick something in the chat or the Q&A.
Oh, yeah, let me see how to find the chat.
Let's see. Yep. Questions for Eric? So I have one for you, Eric. So are you surprised that there's no regional, or that there's only at most a very weak regional pattern to the genetic variation that you've seen looking across species? I mean, I would have thought, especially for things like long runs of homozygosity, that there, where the whole Central Valley has been nuked in the last 200 years. You would really see something strong there. Any comment on that?
Yeah, it's a great question. And again, I'll emphasize that I was really aggregating everything in these analyses. And what, you know, I think there's different ways to parse this. For example, if you were just to pull out the vertebrates, then I suspect we may find something different than just aggregating across everything is probably one answer to that question. And because I think if we were to see a really strong effect at that broad aggregate level, it'd be like the plants, the insects, the vertebrates, they've all just tanked so much that there's been such a strong bottleneck that they're all experiencing rapid inbreeding. And it seems more likely then that variation sort of across the species, most of these species exist across a broad range, in and outside of each ecoregion. That variation is much greater than you see explained by the ecoregions themselves, if that makes sense. So I think, I wouldn't say that I'm going to hang my hat up on like there's not complete regional declines. It's just at least likely species or taxa or clade specific or other aspects of the ecology that I think will emerge from this in a more finer grained analysis. Does that answer your question?
Yeah, to start anyhow. Thanks.
I see a question from Nathan. Is there a way to distinguish non-synonymous deleterious mutations from ones that might result? Yeah, that's a great question. That's a challenge of this broad aggregate across a lot of different projects is doing this reliably. One of the things that the deleterious scores that we calculate are relative to an alignment of proteins in a lot of different species. And so it tends to be things that are quite conserved. And then the ones that we call deleterious are those that are, appear rarely and just in their species of focus. And so it's unlikely that, say, all of them are local adaptation, but we can't necessarily distinguish that from that method. But certainly the magnitude of, say, one population that has many, many, many more than a shared population close to it is probably more likely to be a result of some other process like population decline. But I think that's where bringing in these different metrics can help us understand something about the mechanism that leads to these patterns, if that makes sense.
Oops, sorry. I see a few more questions here, but we probably ought to get a move on. So how about we hold those for the end and move on to, is that okay? Okay. And then move on to Anne, who's going to give another one of our sort of larger talks with Q&A at the end of hers for a few minutes. Anne?
All right, let me share this. Okay. You're looking good. Are we? Okay. All right, great. Thank you all for joining us. This is really exciting to have a session devoted to the CCGP, and I'm excited to talk to you about one of the projects we've been working on kind of the landscape genomics front. So we all know that we're in this era of kind of unprecedented global change, and a key question we have in thinking about how, for example, the maximum temperature of the warmest month is going to change in the next, few decades, we might want to understand how populations are going to adapt, and they need to adapt in order to survive these changes. And so if I show you an example here with one of my favorite CCGP species, this is the Western spadefoot toad. This species occupies areas of the Central Valley and down into sort of more coastal California regions. And we think about the mechanisms through which these populations might be able to adapt to coming change. It's easy to imagine that these populations require some amount of standing adaptive genetic variation. So if they have sufficiently high genetic variation, similar to some of the literature that Eric was mentioning, they will be theoretically able to adapt to these changing environmental conditions. Alternatively, or in addition to this, it's easy to imagine as well that gene flow and the migration of beneficial alleles from other populations into these ones might allow these populations to survive global change, in part because if these neighboring populations have maybe warm adapted alleles that they can introduce into these groups, it will enable them to survive these oncoming changes. And so it's really a question of kind of thinking about both adaptive variation and gene flow, different evolutionary processes, and our ability to understand the extent to which populations will be able to adapt to change, which is known as adaptive potential. And this is the primary focus of this particular project. So ways in which we think about adaptive potential have changed drastically over the years. So initially, we would take a species distribution model. I'm showing you one for our Savannah sparrow, which is one of our CCGP species here. And we would just take the environmental conditions in which this SDM is in and project it forward through time to imagine how the species distribution might shift in accordance to future change. However, there's a lot of assumptions built into this, and this is kind of a simplistic way of thinking about how populations might adapt. And one of the main reasons that it is simplistic, or one of the assumptions it makes, is it's not taking into account anything about intraspecific variation and how different populations might be better adapted to different conditions. So if I show you our population structure results for the CCGP, we can see three sort of distinct population clusters. And if I show you also a map of the annual precipitation across California, it's easy to see that maybe this purple population cluster is better adapted to wetter conditions, and the yellow populations in the yellow cluster are better adapted to hotter, drier conditions. So it's really important for us to think about intraspecific variation in the context of adaptive potential. And the way we do this is using genotype environment association methods. And these types of methods just detect allele frequency changes that are significantly associated with environmental gradients. And what we're left with is something like this, a Manhattan plot on the left, where we can see outlier loci that are significantly associated with our environmental variables. And we might be able to assume then that those outlier loci might confer some adaptive advantage to these populations in surviving their environmental conditions. So if we now look back at our max temperature over warmest months, we can actually take these outlier loci and the adaptive variation contained within them and map this across the landscape. What we end up with is this sort of rainbow map of adaptive composition. And this is showing us the turnover of those adaptive variants. So on the far left, this is a biplot. And if you picture this as just, a method of ordination, we can see each SNP distributed in this space with our environmental variables loaded onto it. And each SNP is colorized, in this case, according to our first redundancy analysis axis. What we can now do is take our future environmental conditions and actually use this information to project it onto that map to get an idea of what the future adaptive composition will look like. And the difference between these two layers is known as genomic offset, which is the change in allele frequencies that's necessary for populations to maintain the present patterns we see of local adaptation. And so this is the typical way that researchers are thinking about measuring adaptive potential. But if we go back to my introduction to this idea and this overall project, one thing we're lacking is gene flow. Thinking about GEA methods and projecting these across the landscape to get at adaptive composition doesn't actually capture the evolutionary process of gene flow and what that might mean for enabling these populations to survive. And so it's really important for us to consider and capture both of those factors. And so that's kind of the main focus of this project, which is to understand how well adapted these populations are in the face of climate change. Our first question is how much variation there is in the first place using those GEA methods and genomic offset. We also want to understand to what degree populations are exchanging alleles through gene flow. And we also want to develop some kind of index of adaptive potential that combines both standing adaptive variation and gene flow. And that's definitely a part of this project that is in progress. So I'm really just going to discuss a couple examples for these first two questions. Before I do so, I just want to briefly mention that, we have a wonderful team of people working on the CCGP. And in thinking about how to get very comparable data sets that are very curated to our analyses and also, it took a lot of careful consideration of our filtering, our parameterizing, and this resulted in us generating a couple pipelines specific for the CCGP. The first of which is our variant calling pipeline, which was developed by Eric and Cade. I think Kate is actually going to talk a little bit about it just in a little while. And then on the back side of things, we've also developed a landscape genomics pipeline that we're using for a lot of these analyses called Alligator. Okay, so first of all, let's take a look at the adaptive composition for one of our bird species in the CCGP dataset. Again, you can see here, the more similar the colors, the more similar the adaptive genetic variation or composition across the landscape. And this is masked to this particular species distribution. When we look at resulting genomic offset scores for this species, we can see that areas around Point Conception and Monterey Bay are kind of flashing as very hot zones, which implies that these are populations that have very high offset, or it's going to require a lot of shift in their allele frequencies at these loci for them to adapt to the future conditions we've input into this model. On the side of gene flow, we've looked at a couple different analyses. The first is population structure. So again, this bird in particular has two population groupings. And what we can now do is take these ancestry coefficient values to look at where there are boundaries or breaks between these population clusters. When we do this, we get something like this. So the redder the color, this means the sort of stronger the population boundary between those two population clusters. And this gives us a rough sense of where we might expect to see breaks in gene flow because there's turnover happening between these clusters. But we actually want to calculate functional gene flow, not just sort of guess at it. And the way we've been doing this is using a method called themes. This is a fast estimation of effective migration surfaces. And Eric has been in charge of running this, so I'm sure he'd be happy to field questions on how it works. But very briefly, our sampling coordinates you can see as these black dots, and these have kind of been snapped into place along this lattice on the landscape. And the lattice, the lines or edges are colorized according to sort of effective migration rate, which is our migration rate relative to expectations of isolation by distance. And so redder or hotter areas represent areas with low gene flow, and cooler blue areas high gene flow. Okay, so we have these metrics of sort of approximated and functional gene flow, and we have our genomic offset maps. Let's overlay them and see what that kind of looks like. So first, let's take a look at just the highest offset regions in this species. So this is the top sort of 99th percentile of our offset values. And if we overlay that on top of underneath our themes map, what we can kind of squint away at is, if I zoom in a particular area here, these are the regions that have our highest offset values for this species. And you can imagine that based on our themes values, these are also areas that are sort of surrounded by barriers to gene flow. And so my kind of takeaway from this for this particular species might be that not only do these populations have very high offset values, meaning they really need to adapt quite a bit to the future change that we're inputting into this, but also they may be limited in receiving beneficial alleles from neighboring populations because of this barrier to gene flow that we're seeing. And so it's really important for us to come up with this metric or index where we can really kind of statistically compare these things, which is something that we're working on. We don't always see this though in some other species. This is another bird. We might see that in, areas of very high offset, again, the coast, but Northern California this time, there's really no breaks in gene flow. And so we would hope that, yes, these populations have high offset values, but they might be receiving hopefully beneficial alleles from other populations that have them. So a bit of an evolutionary rescue type of scenario. Okay. So of course, one of the huge advantages we have for the CCGP data set is that we have a multi-species data set. And we have this opportunity to ask questions across many different taxonomic groups. And to this end, we really want to understand whether there are shared geographic regions in which populations are experiencing high offset, maybe in reference to the California ecoregions that Eric briefly touched on. We also want to understand if there are groupings of species that have similar barriers to gene flow or regions across the state that are consistently breaks to gene flow. And lastly, another aspect of this project we're really interested in understanding is whether the genes themselves that are helping adapt to global change are actually the same among taxonomic groups. So are our ortho groups consistently tied across species to the same, to their ability to adapt. And this has been recently done in various different plant species. So I think a framework exists for us to try to apply it to the CCGP. I'm just going to give you a very brief teaser of the second question. And I'm going to show you again those sort of population boundary maps once again. And this is just a mean of 9 bird species across the landscape. And so again, redder areas are sort of stronger population boundaries, or maybe also breaks to gene flow. And if I overlay the California ecoregions map, what we can really make out here quite clearly is that the Central Valley is really acting as a very strong barrier or an area of population cluster turnover in all of these bird species. If we ramp this up now to 30 terrestrial species, we see a similar pattern emerging, but I would say we also now see the Mojave emerging as well as a barrier. So I think this is just a taste for the types of patterns we're hoping to see across a broad variety of taxonomic groups, and it's exciting to start seeing these sort of patterns emerging. Okay, I want to end there. I'd like to thank Eric, Anusha, our core team members. We also have a landscape genomics working group, which has been wonderful, and members of the Ian Wong Lab have really helped out as well. So happy to take questions.
Any questions? Couple of minutes for Ann? Oh, Raina, hi. You want to just hand?
Yeah, sure. I can read this. Yeah, so should I read the question, or does everyone? Okay. How could you envision the results of the genomic offset stuff being implemented into management decisions for the conservation of these species? This is a great question. I think this is really where the multi-species analyses are going to shine, because I think that for conservation management decisions, we really want to try to work to conserve those corridors of connectivity. So if we have shared areas of high offset, and we can see that there's a potential area of functional gene flow through which multiple species might be able to sort of be rescued in effect, I think we would really want to work to try to conserve or manage those areas that are acting as corridors.
And maybe one more.
Sure. What is your experience regarding depth and sampling size requirements for GEA methods? This is a great question. This is certainly something we've been thinking a lot about. I would say, so first of all, the results that I showed you here, I tailored such that we had really good coverage across the state of about 150 individuals. And I think the key there is for GEA methods, we really need sufficient sampling to capture gradients. We need sampling of individuals across environmental gradients so that we can actually detect those associations. And so it's definitely something we've been thinking a lot about. And I think we are still trying to come up with ways to use the data sets that maybe don't have quite as thorough sampling. We still want to be able to make use of those, but they certainly exist. So it's a bit of a balance. But yes, I think in general, our scale of analysis of 100 to 150 individuals across the state has been sufficient to detect those associations.
Okay.
Yeah.
Yeah.
No, I didn't answer the depth question, but yeah, let's just keep going. I can write it out.
Okay.
Great.
We probably ought to get a move on. And thank you very much, Ann and Eric. And what we now like to do is move into a series of 5 sort of lightning talks. The first 4 will be 5 minutes each, we hope, and the last one will be more like 10. And we'd like to just kind of run through these one at a time. And these are just exemplars of the kind of work that some of our very many constituent labs have been doing and analyzing on their individual projects. So let's get started.
Okay. You want me to take over, Brad?
Yep, take over, Cade.
Let me make sure this is there's
Oh, a little bit of delay. Cool. Yeah. Hi everyone. My name is Cade. I'm a third year PhD student at UCSC. And before I started my PhD, I was working on the CCGP more directly, but now I'm doing stuff on the side. And so I'm going to tell you a little bit about SNP Archer, which Anne already nicely mentioned earlier. Um, so you've got a pile of reads. Now what? Well, whether you are a consortium like the CCGP or you are an individual lab or PI, you're probably going to want to call variants. And as many of you are probably aware, this is easier said than done. This is a common but complicated process. Here I'm showing you GATK's, best practices for variant discovery, which has over a dozen steps. And if you are new to genomics, this is quite a scary picture to look at and quite a high barrier to entry. Even if you've been doing genomics for a while, you are probably familiar with this process, but you know that there's limited support for non-model organisms. In addition, this sort of complicated process introduces lots of variation that can occur in between steps. And these sorts of differences in how you process the data can lead to technical biases that then limit the types of comparative analysis you can do between a data set produced by maybe even one individual in your lab produced compared to another data set produced by a different individual, just based on how you computationally processed your data. And this would limit the types of analysis that you can do. And like Anne and Eric just showed, there's lots of powerful analysis we can do with big data sets, but we need to make sure that they are all processed in the same way. And then finally, reproducibility of this is key. If you yourself can't reproduce what you've done, that's a problem. And then also, you need to make sure that others can reproduce the work that you present in a paper or publication or whatever it is. So the SNP Archer workflow at its core aims to abstract this whole GATK best practices pipeline away from you. And at its core, it needs a reference genome in the FASTA format, and then either your sequencing reads in FASTQ or even reads from the SRA if you're just, you want to mess around with some data that's already publicly available. And then finally, it'll give you your VCF with one command. I'm just going to quickly step through how it does this. It does, your typical read mapping using BWA mem or BWA mem and marking of duplicates and things like that. Then it does individual variant calling using GATK's haplotype caller. And SNP Archer does this in a highly parallelized way. For those that are familiar, they know that haplotype caller is quite a slow step in this process. And so we spent a lot of time making this as quick and efficient as possible. And then, all of these individual haplotype calls can be then joint genotyped, again, using GATK's tools. And this is also done in another highly parallelized way to speed up this slow process. And so this gives you a VCF. And from this VCF, you can start to do whatever analysis it is that you want to do, population, evolutionary things. And SNP Archer also comes built in with some analysis modules that can help you start that, including, quality control things, making genome browser track hubs, and even doing some like MK tests and other things. This is also a room where you could develop your own module if you want to contribute it back to the SNP Archer ecosystem, which is something that we are pushing heavily. And so for now that you can, this whole map is available, or you can see this whole map now. Some people, again, if you're newer to genomics, this might be kind of scary. If you've been doing it for a while, you are familiar with this. But the whole point of SNP Archer is that it abstracts that away from you. So if you don't want to mess with any of the details, you don't have to. But if you want to, you can go into the workflow and modify things as you see fit. I just want to quickly highlight one of our modules, which is the interactive QC dashboard that Eric, who presented earlier, led the development of. And so this takes your VCF and subsamples it, and then performs some common analyses to highlight any maybe odd samples or technical noise that might be present in your variant data set. It does this with, performing PCA, doing comparisons of depth and missingness, as well as highlighting any maybe related samples that you might want to drop from your downstream analyses. And that's all I have. Thank you for listening. I'd like to obviously acknowledge the whole SNP Archer team, all my co-authors, as well as the CCGP team who were instrumental in helping get this project off the ground. You can check out our paper with this QR code. I guess maybe you probably can't see my cursor. And then there's, you can check out our Git repo with that other QR code. And this is all still in active development. We're working on some really big upcoming features and changes to make this even easier to use. If you have used it, we'd love to hear from you. You can email me or drop an issue on GitHub. And yeah, thank you.
Okay, thanks a million, Cade. That was great. And we're now, as I said, just going to move on to John Richmond.
Hi there. So Courtney, you're going to advance the slides, right? Just assume you are. So hi, good afternoon. I'm John Richmond. I'm a wildlife geneticist with the USGS here in San Diego, California. And my talk just glazes over one of the target projects of reptiles in the CCGP, which includes two closely related species that are in the same species complex, and the bulk of their distributions occur in California. And so the resequencing part of the work, the 150 range-wide samples that Brad referred to as the heart and soul of the CCGP, hasn't actually been completed for these species. So this talk's more of a wish list for what we can do with the data beyond its core goals that he outlined earlier. But that said, go to the next slide. The reference genome has been sequenced for the Gilbert skink, Plesion gilberti, which is one of the 4 species within the complex. The total genome length is about 1.5 gigabases, which is on the shorter end for lizards. But I included this slide just as an advertisement for Brad that it's been done. So don't break your eyes trying to read it at all. But on the left is the snail plot that just summarizes a few metrics about the sequencing quality and contiguity, like the scaffold and 50 score, which was 184 megabases, the BUSCO completedness scores for both the primary and alternate genomes, which were 89.3 and 97.2% complete. And then you see the contact max for both of those genomes on the right. Some other things, the read coverage is great at 56.1x, and the sequencing error rate was like 0.2%. So all that's to say that the CCGP has generated a high quality reference genome for this species. The next slide. So all members of the species complex are united by this shared ancestral ontogeny where the juveniles have these brightly colored tails and stripes that make them really hard to catch and eat. And so here I'm showing the juvenile phenotypes above the horizontal lines and the adults are below. And then on the left and right sides are representatives of 2 predominant ecomorphs. The three species are categorized as small and one's large, with that large body Gilberti morph reaching about 1.5 to 2 times the size of the small morph, and then transitioning in color as individuals age. There's some limited sympatry between the two, but the larger Gilberti are found in more xeric environments compared to the smaller species, which occur in more mesic environments. And so for reasons I'll touch on next, we'd like to use a CCGP to learn something about the genes that actually underpin these size differences and the ontogenetic shifts in color pattern, and also whether there might be functional genes tied to other traits that could also be increasing fitness in these different environments. So slide 4. So for this one, back in the late 90s when music was good, mitochondrial DNA was the gold standard for phylogeographic studies. So on the left, I'm showing a range-wide mitochondrial phylogeny. And what that told us at the time was that the large-bodied morph wasn't a single species, but instead had 3 separate origins. So the black tree branches are all haplotypes belonging to the small ecomorph populations. And in the tree, we see pairs of reciprocally monophyletic ecomorph lineages, which suggested that the same reproductive isolating mechanism had evolved multiple times independently, so something referred to as parallel ecological speciation. And so that led me to experimental work that then tested whether ecologically important traits, namely body size, were tied to reproductive isolation. And the short story is that too much body size divergence disrupts the geometry of this pretzel-like mating posture that you see on the right. So we had an ecologically divergent trait that's acting to mechanically prevent gene exchange between ecomorph populations. So there's a ton I'm leaving out, but the CCGP data, again, is going to allow for more rigorous testing of the parallel speciation hypothesis, and 2, identification of the genetic mechanisms involved with this size divergence. So next slide. We have collected some RAD-seq data in recent years, and they present a contrasting story with the mitochondria. but it's not terribly surprising, and the differences are predictable. So here I'm showing a tree that was estimated from concatenated rad loci with the branches colored according to the individual's cluster assignment. So there's 9 clusters in the vertical admixture plot that line up with the tree, with the map on the right showing the ancestry proportions as pie charts. And with these data, we no longer see parallel morphotype evolution, which was sad initially, but I'm in a happy place now. So what we think happened was that there were 2 separate instances of ancient hybridization in these areas indicated by the arrows on the map, where in both cases you have the mitochondria of Sciltonianus crossing species boundaries into Gilberti and then diffusing throughout more than 3/4 of the range of Gilberti. But the interesting thing is that despite this widespread integration of foreign mitochondria and Gilberti, there's almost no indication in the genomic data that the 2 species have ever hybridized. So the CCGP data is going to help us understand what the fitness gains might be to acquiring foreign Sciltonianus mitochondria in Gilberti over most but not all parts of its range, and then knowing whether certain parts of the nuclear genome might have introgressed to a greater extent than others, and if so, why that might be. For example, do those introgressed parts contain loci that are under selection, or are they linked to loci that are under selection? Then 5 or 6, the last slide. I'm really fortunate to be working with a postdoc in Ian Wong's lab at UC Berkeley, Dr. Ben Karen, he has an NF grant to study the genomics of tail color in these skinks. And for all the projects I've just mentioned, he'll be involved with them if things go my way. He's this great guy, super smart, and he loves skinks. So juvenile tail color is like, it's the trait these skinks are known for. And with Gilberti, depending on location, they either have these pink or blue tails. And so on the right, Ben has mapped tail color onto the CCGP sampling, and notice the blue circle where you see these kind of like frozen dessert-like colors in the tail color. And from our RAD data, we know that this is an area of secondary contact with gene flow between divergent blue and pink-tailed lineages of Gilberti. So the questions are, what caused this shift from blue to pink? What are the genetic underpinnings of the 2 different colors? Is it something simple, like a single gene of major effect, or is it something more complicated? And then could integrate tail color be a post-zygotic isolating mechanism because it's less effective as a decoy than true colors? Or does it even matter if it's pink or blue or integrades? And so these are the 3 things that Ben wants to do with CCGP genomes to get at these questions. And because I'm super excited about what he wants to do with all this, I'm going to stop here to avoid rambling. So grateful to be a part of this project. Thank you for listening.
Thank you very much, John. Appreciate it and appreciate the skinks. Let's move on to Kim and bumblebees.
Hi, everyone. My name is Kim Ballar. I am a research ecologist with the U.S. Forest Service, but this work was primarily done when I was a postdoc at UCSC in Beth Shapiro's lab, and was done in collaboration with entomologists Neil Williams and Hollis Woodard. So I'm going to describe just kind of the genome assembly rationale and process for 2 bumblebee species that we worked with. So as you may or may not know, bumblebees are in decline worldwide, with up to 1/3 of species at risk of extinction. And genomic information is crucially needed to inform this conservation. Some bumblebee species are stable or potentially increasing, though, and reasons for this disparity between species is somewhat unclear. And we think that genomes could provide clues and inform conservation. And conserving these species is really important because bumblebees are really important pollinators for both wild plants and many commercial crops. So there's currently somewhat limited genomic information of bumblebees. We have 40 total species genomes at various levels of quality on NCBI, with 13 North American species currently. And so we focused on 2 species that are relatively historically widespread in California. So the first is Bombus sonoris, or the Sonoran bumblebee. And we think this is likely in decline based on recent sampling. It's also a potential conspecific to the American bumblebee. They're Bombus pennsylvanicus, which is an eastern subspecies. There's some taxonomic debate between those, so genomic information could also inform that. But there are no genomes available for this species previous to ours. And then in contrast, we also sequenced Bombus vosnensensky, or the yellow-faced bumblebee. This is the most widespread bumblebee in California. If you live in California and you see bumblebees, probably this one. And population is stable relative to other species. But I am also showing some species distribution modeling for both species with historic distribution on the left in the map and then the more current distribution on the right. And you can see that for Bombus sonoris, the range is pretty severely restricted to Southern California now. And for Bombus vosnensensky, it's still pretty widespread, but there is some restriction there. And it is also actually an important model species for molecular ecology research, the yellow-faced bumblebee. So recent analyses have shown that there is some reduced heterozygosity population-wide for this species. So I think that it's important to more fully understand genetic diversity for both species to inform their management. So what we did was we collected wild specimens from Southern California for both species, and we followed the standard CCGP pipeline for library preparation and assembly methods. And we did return, both species returned contiguous genomes with pretty low numbers of scaffolds and high BUSCO scores. And luckily, as a sanity check, they both returned about 18 chromosome length scaffolds for each, which aligns with the typical Bombus karyotype of 18 chromosomes. And we also did some genome comparison with a few close relatives that were available on NCBI. And so here I'm just showing a genome alignment plot between our assembly of Bombus sonoris on the x-axis, and then an annotated assembly for Bombus dalbaumii, which is the most closely related species to Bombus sonoris available on NCBI. And so each of these little boxes here is just an alignment between the annotated chromosome and one of our scaffolds. And so you can see I'm highlighting here in red boxes some incongruencies we found. You can see that the genomes are relatively well aligned, with most of the scaffolds having pretty long forward alignments with the other genome, but we do see some reverse alignments that highlighted in green that aren't really quite matching up with the other genome, and we're not really sure what's causing this. I just wanted to show this just as something interesting that we might investigate in the future to see what's going on. And so our next steps, I'm hoping to submit the genome announcement manuscript any day now, just waiting for some co-author feedback. We're waiting for the chromosome annotation to get done in NCBI. And then ultimately the goal, as for all CCGP projects, is to do some landscape genomic analyses for all these species. And these reference genomes will also serve as references for conservation analyses of 4 other bumblebee species that are, I think almost all of them are probably in decline in California. So this will be really important to inform their conservation. So with that, I want to acknowledge my collaborators on this project and of course the CCGP for funding everything. Thank you.
Great. Thank you, Kim. And next up is Nathan Ray.
Hello, everyone. My name's Nathan Rank, and I'm from Sonoma State University. And the work I'm going to talk about today was conducted in collaboration with researchers at UC Berkeley and Santa Clara University. We study a leaf beetle that is adapted to cold conditions and lives in California, but most species in its major lineage occur a lot further north in North America and Eurasia. So I've been studying this species in Sierra Nevada for a long time, and been studying the evolutionary ecology. And some features of it are that it completes its life cycle only once per year, regardless of where it's found. So actually, individuals spend a lot of their time in dormancy, overwintering. The insect lives only on willow species, so it's restricted to environments where willows grow, so bogs and streamside environments in relatively cool areas within what is mostly a pretty warm state. The populations that I study in the mountains experience pretty extreme fluctuations in temperature on a daily and seasonal basis, and they also live under reduced oxygen supply that affects the components of fitness. So for the CCGP, we've compared populations in 3 different major regions of California. As you can see, a lot of the range is within the Sierra Nevada mountains and the eastern, sort of central eastern part of the state. There are some populations found in the northeast corner of the state, also in a montane region where the Sierra Nevada mountains meet the Cascades, and then some populations north of San Francisco along the coast. And what these populations all share is that they occur in cool, moist regions that are separated by substantial areas that are warm and dry. So for the CCGP, we got genomic data for about 30,000 protein coding genes and the nuclear genome. And then we also looked at about 15,000 base pairs that constitute the coding region of the mitochondrial genome. So the results of the nuclear study are that there was a lot of variation within the Sierra Nevada mountains, even though it's a relatively small portion of the range in California. You can see from this principal components plot that there is a lot of divergence between populations along a latitudinal gradient in the Sierra. And populations in the Sierra are quite distinct from those in the northeast corner of the state and the other montane region. And both of these sets of populations are distinct from the ones in the coastal regions north of San Francisco. If you look more closely at the individual level using the structure plot, you can see that the results are consistent with what I described a minute ago. Coastal populations very distinct from northeastern ones, which again are distinct from the Sierra Nevada ones. But you can also see that within the Sierra Nevada, it looks like there are 3 major lineages ranging from south near Tuttle Creek to Rock Creek in the north. And there are a couple of drainages that show a lot of evidence of interbreeding with individuals from, or at least a lot of intermixing of populations from adjacent drainages further north. So you can see that in Taboose Pass in the southern part of the range and in Bishop Creek in the central Sierra. When you look at the mitochondrial genome, it looks as though it's pretty consistent with what I just described, with fairly distinct populations in the northeast corner, as well as in Gualala River. And then the distinct lineages within the Sierra Nevada, which also show those regions like Bishop Creek and Taboose Pass with some degree of mitochondrial variation within the populations. We looked at the genetic divergence between the coast and the mountains, and we found evidence overall for isolation by distance, but we also saw that populations along the coast are pretty distinct from those in the montane regions, indicating that there may be adaptive variation showing up here. That we also looked at some loci that are specifically associated with snowfall and air temperature using some of the methods that were described earlier. And so we've identified several genes that may well be involved with adaptation to variable winter conditions. And here's a picture of the individuals who worked on this project as well as some other projects. And you can see overall, again, that we found that mitochondrial nuclear genes vary greatly among populations along the latitudinal gradient, that we have some hotspots of genetic diversity within the Sierra Nevada where populations are interbreeding, and we've got some evidence of adaptation to winter climate within California.
Thank you, Nathan. It's great to see. As you say, working on the system for a very long time, and it's very cool to see it tied up or getting tied up. And our last is Anusha Bishop, and Anusha, take it away.
Thanks, Brad. So my name is Anusha, and I'm a graduate student at Berkeley, and I'm also on the landscape genetics team for the CCGP. And I'm really excited to talk with you all today about some of my work on the landscape drivers of genetic diversity across California, specifically in western fence lizards. So we all know that genetic diversity is really important. The loss of genetic diversity can have consequences in the form of inbreeding, which affects long-term population viability. And genetic diversity also provides the foundation upon which evolution can act, specifically for species, in responding to new stressors. And protecting genetic diversity is inherently a spatial problem. So understanding how genetic diversity varies across space can allow us to protect valuable areas of high genetic diversity, identify vulnerable areas of low genetic diversity, and overall determine the drivers of genetic diversity patterns. So what exactly drives genetic diversity across space? Well, fundamentally, landscapes drive variation in population size, gene flow, and selection through time. And cumulatively, these processes are what shape the patterns of genetic diversity that we see today. So we might expect that areas that have historically been more stable are going to have greater genetic diversity, while areas that have experienced, population bottlenecks or range expansion might have lower genetic diversity. And we often think of these processes as occurring over very long periods of time, so maybe glacial cycles over hundreds of thousands of years. However, recent research has also shown that contemporary landscapes, human impact, recent climate change, have the potential to shape genetic diversity patterns. But we still know relatively little about the roles and relative contributions of all these processes, both historical and contemporary, to shaping the genetic diversity patterns that we see today. To kind of dig into these questions, we're going to go into California, which is a great place to study landscape variation because there's a lot of landscape variation, both in historical and contemporary processes. So, for example, past paleoclimate stability through the glacial cycles, human modification, recent climate change, just the landscape in general, vegetation, temperature. And the hero of our story is Scalopris occidentalis, also known as the western fence lizard. And there's these small generalist lizards found almost everywhere in California, so across a wide range of habitats, elevational gradients, basically everywhere you look in California, you're probably going to see a Scalopris. And Scalopris are also a great species to study these processes because they share a very long history with the California landscape. Early work by Bouzid et al., published in 2022, estimated the initial divergence time of about 1 million years ago in Southern California, and then showed that there's probably this northern range expansion over hundreds of thousands of years throughout the Quaternary glacial cycles. So to dig into what the drivers of genetic diversity are in Scalopris, we're using whole genome data from the California Conservation Genomics Project for 162 individuals across California. And for each of these individuals, we calculated genetic diversity based on heterozygosity across approximately 14 million SNPs. We then modeled genetic diversity as a function of both historical and contemporary landscapes. So for historical landscapes, we included things like paleoclimate temperature change, so temperature change over the last 100,000 years. We also included factors like bioclimatic stability, which captures environmental stability over much longer periods of time, all the way back to the Pliocene, as well as last glacial maximum glacial extents. And then for our contemporary landscapes, we included variables like recent temperature change over the last 100 years, current temperature and vegetation, and human modification. Since this is a lightning talk, I don't really don't have time to go super into how we did model selection and all these other variables, but I just want to highlight one really cool result that we found, which is that paleoclimate change is the strongest predictor of genetic diversity. So here on the right, we have a map of all of our individuals colored by paleoclimate temperature change at each point. And then on the left, we have a map of plot of paleoclimate temperature change versus heterozygosity. And we see a really, really strong relationship between paleoclimate temperature change and heterozygosity. Like this is explaining 20 to 30% of the variance overall in whole genome diversity, which is really cool to see. And this might make sense. Remember, we talked about there's this potential northern range expansion. And we think that maybe range expansion and population size fluctuations throughout the quaternary glacial cycles, it's what's driving this pattern. But what about contemporary landscapes? What's tricky is that we do see significant associations between contemporary variables such as contemporary temperature, recent temperature change, and human modification and genetic diversity. However, these variables often don't end up in going through our whole model selection procedure because they're significantly associated also with paleoclimate change. So it's really hard to disentangle these historical and contemporary landscape drivers because they're correlated. But this doesn't mean that we should just discount contemporary variables as playing a role in the genetic diversity we see today. It means that we really need to make sure to be including both historical and contemporary variables when we're trying to assess the drivers of genetic diversity. So we finally were curious what this might mean for the future. We were thinking, we know past climate and stability has impacted the genetic diversity patterns we see today. What does this mean about how future climate change and current genetic diversity might interact? So specifically, do we expect that areas of lower or higher genetic diversity may experience less or more climate change in the future? And we see a really strong relationship between future climate stability and genetic diversity. Specifically, we see that future climate stability is lower in areas of low genetic diversity. So here on the x-axis, we have climate stability, which is based on the bioclimatic stability index, which captures how we expect temperature and precipitation stability to happen over the next 100 years or so. And this is for different scenarios of increasing climate severity from left to right. And then we have current heterozygosity that we see today on the y-axis. And so you see this positive relationships, which means that areas that are going to be less stable in the future have lower genetic diversity, and areas that are more stable in the future have higher genetic diversity. And this might have consequences for adaptive responses to climate change. The main takeaways from this work is that paleoclimate change best explains the genetic diversity patterns that we see today. However, we can't disregard contemporary landscapes as well because of these confounding correlations between past and current landscapes. We also see evidence that future climate change is going to disproportionately affect areas of low genetic diversity due to the role of past landscapes in shaping genetic diversity patterns. So our next steps are to look at genetic diversity through time. So using methods like PSMC and SMC to see if changes in effective population size through time also align with these glacial cycles. We also recently got an annotated genome for this species, which is going to allow us to look at adaptive variation and local adaptation and ask questions of how these patterns in genome-wide genetic diversity might affect adaptive potential. And finally, as part of my work with the CCGP, we're going to be expanding these analyses to the whole CCGP dataset to ask these same questions across the tree of life and identify common drivers of genetic diversity across California. And with that, I'd like to acknowledge all my collaborators and funders, and thank you all for coming. I'm happy to take any questions.
Okay, thanks, Anusha. Thank you very much. Let's see. Okay, so of course, we said we were going to be done and ready for questions at 12, and it's now 12:24, so we have 6 minutes for questions, which is, which is great. I think it was a wonderful set of talks, and what I hope it gives our participants a flavor of is both the analytical side of what we've accomplished. Remember, many of those analyses and approaches that Eric and Ann and Cade discussed, those are new. Those are things we developed in the course of this project, and also our empirical approaches, the small handful of case studies. Those are individual case studies, but also, we can pull them together into where the real power of the project lies, as Ann said, in comparative approaches. So do we have questions from folks who are still online? We've got a couple, few minutes, could take a couple, 3 questions for anybody or everybody. And if you do, I'd say just either stick it in the Q&A or the chat, or speak up if we can make that happen. Any questions? Questions? No. Not seeing anything there. Anusha, I want to ask you a quick question, since of course I care deeply about Scalopris. So if historical and contemporary are confounded because they're sort of they're similar in their effects, why do you give it all to historical?
That's a great question, Brad. Yeah, because it's something I've been thinking about a lot. I think it's a combination of things. I think one, the effect of the past is much stronger. 2 is we have this knowledge from other analyses, from Bouzid's work on, the divergent states for the species, and then some kind of work that I'm doing right now using PSMC to kind of mark changes in population size with more deep time processes. But I think this is a good question. I see a lot of papers that look at one variable and not the other. And then it's like, well, we don't really know, because these are all correlations.
Right.
Which is the stronger one. So I think this is where the biology of the species matters. This is where also other analyses that complement the kind of spatial analyses, but with temporal stuff to really mark those two, becomes important.
Yep. Okay. Any other questions that people would like to ask our speakers? Okay, and we have been on for quite a while, so maybe it's that time. So Courtney just put in, a link to our website, and hopefully if you liked what you heard, if you want to start your own, take out the first C, and make it the Nevada, conservation project, or Florida or Texas or whatever it is you might want to do, please get in touch with us. This has been it's been an amazing ride and an amazing journey, which we'll be finishing up within the next year. And it would be wonderful if it stood as a model for other states, other regions, other, whatever, to be able to bring genomics into the toolbox of how we manage wildlife, how we manage natural resources in the US and abroad. And it would be wonderful to talk to you about it, to help plot and plan and see that happen. So I think we're about at time. And so I want to thank everybody. I sure as heck want to thank the speakers. And for some great presentations and great insights, and thank the many people who joined us for sticking around. Thanks a lot. Okay, why don't we call it? Bye-bye.